Right now, in the time it took you to read this sentence, your body has already started making decisions it wasn't making an hour ago. Not decisions you're aware of, not decisions you asked for. Somewhere between your last meal and this exact moment, a chain of biological events began quietly rewriting how your cells get their energy, how your liver spends its reserves, and, as strange as this sounds, how your immune system decides which of its own cells deserve to keep existing. Over the next 48 hours, that chain doesn't slow down. It builds. And by the time we reach the final hour of this video, you'll understand exactly why scientists keep coming back to 48 hours as one of the most biologically significant windows in all of fasting research. Most people assume that fasting begins whenever they decide it begins, the moment they close the kitchen cabinet, or the moment the clock hits a certain hour. Biologically, that's not quite true. Your body runs on its own internal accounting system, and it starts adjusting long before you'd ever describe yourself as fasting. Within about 1 hour of finishing a meal, blood glucose levels peak and then begin to decrease as tissues either burn that glucose directly or convert it into storage forms of fuel. This isn't dramatic. It isn't something you'd notice sitting at your desk, but it's the very first domino in a sequence that will eventually touch nearly every organ system in your body. By around 2 hours after eating, something more decisive happens. Blood glucose returns to its normal fasting range, and that drop signals the pancreas to pull back on insulin secretion. Insulin is often described in oversimplified terms as the fat storage hormone, but its actual role is closer to that of a traffic controller, deciding whether cells absorb glucose for immediate use or whether the liver should be storing it away for later. When insulin drops, that traffic pattern reverses. The body stops being told to store and starts preparing quietly to release. This is where the liver enters the story, and it enters early. As blood glucose declines and insulin falls, the liver responds to that hormonal signal by beginning to break down its own glycogen reserves, releasing glucose back into the bloodstream, a process called glycogenolysis. Glycogen is essentially your body's short-term savings account for carbohydrate energy, built specifically for moments exactly like this one. What almost nobody realizes is that this withdrawal from the savings account starts within hours of your last bite, not days. The clock most people imagine, one that starts ticking sometime tomorrow, actually started before dinner even finished digesting. By the 4-hour mark, the body has fully transitioned out of what physiologists call the fed state and into something distinctly different. Fasting, in the physiological sense, is generally considered to begin somewhere between 2 and 4 hours after a meal, once blood glucose has settled back to baseline levels. It's a strange thing to sit with. The idea that you're technically fasting for the majority of any ordinary day, simply in the gaps between meals, without ever labeling it that way. The liver's glycogen reserve isn't infinite, and it isn't meant to be. Any glucose your body doesn't use immediately after eating gets stored in the liver as glycogen, and once that food-derived fuel runs out, the body activates a series of hormonal and metabolic responses that convert liver glycogen back into usable glucose. Think of it less like a light switch and more like a dimmer. A gradual handoff from food just arrived energy to stored reserves energy, timed by hormone levels rather than the clock on your wall. What makes hour 4 particularly interesting is that this is where the body begins actively rationing rather than simply reacting. Glucagon, insulin's biological counterpart, starts rising as insulin continues to fall, and that shifting ratio is what actually drives the liver's glycogen release. Not hunger, not willpower, not anything you consciously control. It's a hormonal handshake happening entirely beneath awareness, and it's the reason the next several hours matter so much for everything that follows. The liver, at this point, has effectively started a countdown on its own reserves, and that countdown has a fairly predictable endpoint. 12 hours in, roughly the length of an overnight fast, the body crosses into what researchers call the basal, or postabsorptive, state. A person is generally considered to be in this basal state after an overnight fast, once no food has been eaten since the previous evening's dinner, and by this point serum insulin is low while glucagon is on the rise. This is the exact metabolic snapshot captured every time someone gets a fasting blood glucose test at a doctor's office. A detail that connects a familiar clinical routine to a much larger biological story most people never hear. This is also where the liver's glycogen reserves start running genuinely low, not just being tapped. According to a landmark review by physician researcher George Cahill, whose decades of work on human starvation metabolism remain foundational in this field, 12 to 24 hours of fasting in humans typically produces a 20% or greater decrease in serum glucose alongside depletion of hepatic glycogen stores, accompanied by a broader metabolic shift. 20% might sound modest, but for a system that depends on tightly regulated glucose levels to keep the brain functioning normally, that's a meaningful move, enough to force the body toward an entirely different fuel strategy. And that's precisely the crossroads. With glycogen running low, the body has to find glucose somewhere else, or shift toward a different fuel source entirely. Physiological research on prolonged fasting describes this pattern clearly. Carbohydrate reserves like glycogen are depleted first, and then, within the first 24 to 48 hours, gluconeogenesis increases, drawing on amino acids and glycerol to manufacture new glucose. In other words, by hour 12, the body isn't just spending savings anymore. It's beginning to manufacture new currency out of raw materials it wasn't originally planning to use this way. Somewhere around the 16-hour mark, a shift happens at the cellular level that has nothing to do with blood sugar directly, and almost everything to do with how individual cells decide to spend their energy. Inside cells throughout the body, a signaling pathway called mTOR, which normally promotes growth and construction of new proteins, starts quieting down as nutrient availability drops. As mTOR activity decreases, a different pathway called AMPK, which functions as the cell's internal energy sensor, begins to rise in its place. AMPK behaves almost like an alarm system, signaling that resources are becoming scarce and that efficiency now matters more than growth. One of AMPK's most important downstream effects is switching on autophagy, the cellular process by which a cell breaks down and recycles its own damaged components, including old proteins and worn-out cellular structures. This isn't a fringe theory or a wellness industry talking point. It's a well-documented cellular maintenance mechanism, and it marks a genuine shift in priority, from a cell that's building new material to a cell that's cleaning house. The interesting part is that this switch doesn't require anything close to true starvation to begin. Even a moderate, ordinary fasting window is enough to start tipping that internal balance. Some of the clearest evidence for how powerful this switch can be comes from animal research rather than human trials. In a study conducted on mice at the Whitehead Institute and MIT's Koch Institute, published in the journal Cell Stem Cell, researchers found that a 24-hour fast prompted intestinal stem cells to shift their metabolism away from carbohydrates and toward burning fat, and that this fatty acid oxidation switch measurably improved the stem cell's regenerative function in both young and aged mice. It's important to be precise here. This particular finding comes from a mouse model, not human subjects, and the researchers themselves noted it as a discovery about mouse intestinal biology specifically. But it offers a vivid, concrete picture of exactly the kind of cellular rewiring that begins, in a more gradual and less dramatic form, somewhere around this point in a human fast as well. The 24-hour mark carries a strange kind of biological contradiction, and it's one of the more counterintuitive moments in this whole timeline. While the cellular signaling pathways that drive growth and construction are quieting down, one particular hormone is doing the exact opposite, surging upward in a way that seems, at first glance, to make no sense at all. That hormone is growth hormone, and its behavior during fasting has been documented since the late 1980s. In a study conducted by endocrinologist Kwok Ho and colleagues, published in the Journal of Clinical Investigation, researchers tracked growth hormone secretion patterns in 6 healthy men across a control day of normal eating and the first day of an extended fast. They found that fasting significantly enhanced growth hormone secretion, amplifying the hormone's naturally pulsatile release pattern through changes in both how frequently it was released and how much was released with each pulse. More recent human research has echoed this finding with striking numbers. A secondary evaluation of a randomized controlled trial, drawing on blood samples taken at the start of a fast at 24 hours and again at 48 hours, documented marked increases in growth hormone over the first 24 hours of water-only fasting, changes that occurred independently of any weight loss, and were most pronounced in people who started with lower baseline growth hormone levels. So why would the body ramp up a growth-promoting hormone at the very same moment it's dialing down growth-promoting cellular signals? The apparent contradiction resolves once you understand what growth hormone is actually doing here. It isn't stimulating new tissue construction the way it might during childhood development. Instead, it's protecting existing lean tissue, mobilizing fat for energy, and helping preserve muscle protein while the body leans harder on fat as fuel. In other words, at hour 24, the body isn't confused about whether to build or clean. It's doing something more sophisticated, using one hormone to safeguard what already exists, while cellular maintenance processes elsewhere handle the cleanup. Here's where a popular misconception deserves some correction, because it's one of the most commonly repeated claims in fasting content, and it isn't quite accurate at the 48-hour mark. Many videos on this exact topic imply that by day 2, your brain has already switched over to running primarily on ketones. That's not what the research actually shows for this window of time. In the fed state, the brain relies almost entirely on glucose, and during fasting, it gradually begins co-utilizing ketone bodies alongside glucose. But this is a gradual co-utilization, not a wholesale switch. The finding that ketones can eventually replace glucose as the brain's dominant fuel source comes from a landmark 1967 study by Owen and colleagues, published in the Journal of Clinical Investigation. And it's worth being precise about what that study actually measured. Researchers catheterized cerebral blood vessels in 3 patients undergoing 5 to 6 weeks of continuous starvation, finding that beta-hydroxybutyrate and acetoacetate had, by that point, replaced glucose as the brain's predominant fuel. 5 to 6 weeks, not 48 hours. At the point this video is describing, ketone levels are genuinely rising. That part is accurate. But the brain is still leaning heavily on glucose supplied through gluconeogenesis, the liver's manufacturing of new glucose from amino acids and glycerol. The dramatic headline-friendly version of "Your brain now runs on fat" belongs to a much later chapter of extended fasting, one measured in weeks rather than hours. What's actually happening at this stage is quieter and, frankly, more interesting. The brain is being gradually introduced to an alternative fuel source, sampling it in small amounts, long before it ever needs to depend on it completely. By hour 36, something begins happening to the immune system that most people have never heard connected to fasting at all. A process that researchers describe as a kind of temporary demolition before reconstruction. The clearest evidence for the earliest stages of this process comes from animal research. In a study examining T cells specifically, researchers found that a 24-hour fast in mice increased T cell counts in the bone marrow, while simultaneously reducing them in the blood, spleen, and lymph nodes. and this redistribution did not appear to be caused by cell death, since the fasting period didn't change certain proliferation markers. It's important to note clearly that this specific finding comes from a mouse model, not human subjects. A separate line of animal research examined a different immune cell type entirely, monocytes, which are produced in the bone marrow and circulate through the body fighting infection. In mice studied through several hours of fasting, researchers discovered that monocytes traveled back into the bone marrow to essentially hibernate, while the production of new monocytes in the bone marrow simultaneously slowed down, and the cells sheltering in the marrow survived longer and aged differently than the ones that remained circulating in the blood. This, too, is mouse model evidence, though it maps onto a broader pattern that researchers have started documenting in human contexts as fasting periods extend further. That human dimension comes primarily from work by biogerontologist Walter Longo and colleagues, whose research included both mouse experiments and an early-phase human clinical trial. Their results showed that prolonged periods of fasting significantly lowered white blood cell counts, and in the mouse portion of the research, this appeared to shift the signaling pathways of hematopoietic stem cells, the cells responsible for generating the blood and immune system, from a dormant state into one of active self-renewal. It's worth being precise about the timeline here too. This human data comes from multi-day fasting protocols, generally spanning several days rather than exactly 36 hours. What's happening at the 36-hour mark, based on the animal evidence available, appears to be the opening phase of that same broader pattern. Older or less necessary immune cells beginning to withdraw from circulation, setting the stage for a more complete cycle of renewal that, in humans, tends to unfold more fully over the days that follow this particular fast. There's a specific worry that surfaces in almost every conversation about fasting. Is this eating away at my muscle? The honest, evidence-based answer at the 48-hour mark is more reassuring than most people expect. And it comes down to a remarkably elegant piece of biochemistry that's been understood since the 1970s. Research examining the phases of fasting physiology, building on foundational animal and human work by researchers Lignot and Lamajo, describes an early phase covering roughly the first 24 to 48 hours, during which glycogen stores are depleted and the body draws on a mix of fat and protein for fuel, in a ratio of approximately 70% fat to 30% protein. That 30% isn't nothing, and it's exactly why some muscle protein breakdown is happening right now, even without any noticeable change in strength or how you feel. But here's the crucial detail that context often leaves out. This pattern shifts fairly quickly toward a dedicated protein-sparing mechanism. One, the body activates specifically to slow this early rate of muscle breakdown as the fast continues. The specific pathway responsible for handling this early muscle protein use is known as the glucose-alanine cycle, sometimes called the Cahill cycle, after the same researcher whose starvation studies have appeared throughout this timeline. In this pathway, amino acids released from muscle protein breakdown are converted into alanine, which travels through the bloodstream to the liver, where it's used both to dispose of nitrogen waste through the urea cycle and to generate new glucose through gluconeogenesis. It's a genuinely clever recycling system. Muscle tissue isn't simply being sacrificed for fuel. It's being used in a controlled and limited way as a temporary raw material supplier for the liver's glucose-making operation, one specifically designed by evolution to taper off as the body shifts more fully toward burning fat. By the time 48 hours arrives, none of the individual systems this video has covered are operating in isolation anymore. They're converging, each one influencing the others in ways that make this specific window genuinely distinct from either the first few hours or a much longer multi-day fast. Liver glycogen, largely exhausted since somewhere around hour 18 to 24, has handed the baton fully to gluconeogenesis and rising fat oxidation. Growth hormone, elevated since the 24-hour mark, continues protecting lean tissue, while fat mobilization intensifies. And the protein-sparing mechanism described a moment ago has now had a full day to establish itself, meaningfully slowing the rate of muscle protein use compared to where it stood at hour 24. This is also the point where the phase model described by fasting researchers marks a genuine transition. The early phase, characterized by that initial 70-30 fat-to-protein fuel mix, gives way to a second phase in which the fuel mix shifts decisively toward lipids, with the protein-sparing mechanism now fully established, a phase that, in humans, can extend for weeks depending on a person's starting body fat. 48 hours, in other words, isn't just an arbitrary point on a calendar. It's close to the actual biological hinge between two genuinely distinct metabolic states. What makes this moment particularly worth pausing on is how much is happening beneath a surface that, from the outside, looks completely unremarkable. Someone at hour 48 of a fast doesn't look different. They may not even feel dramatically different, depending on the person. But internally, their liver has switched fuel production strategies. Their hormone profile has shifted in ways that would have seemed contradictory without this specific context. Their muscle tissue has activated a genuine protective mechanism. And, as covered earlier, the earliest signals of immune cell reorganization are already underway. 48 hours turns out to be less a single event and more a convergence point, where several independent biological clocks briefly line up. Stepping back from the hour-by-hour detail, there's a bigger question worth sitting with. Why does the human body have any of these mechanisms at all? The answer connects to something anthropologists and physiologists have long pointed to. For the overwhelming majority of human evolutionary history, reliable daily meals were not a guarantee. Periods without food, sometimes lasting days, were a routine feature of life, not a rare emergency. The systems this video just walked through, glycogen mobilization, the alanine cycle, growth hormone elevation, the gradual ketone transition, the beginnings of immune cell reorganization, aren't malfunctions or stress responses in the negative sense. They're a coordinated, evolved toolkit, refined over an enormous span of time for keeping a brain and body functioning through exactly this kind of gap. What's striking, looking back at everything covered here, is how little of it resembles the popular, oversimplified version of fasting that circulates online. There's no single dramatic switch that flips at hour 16, or hour 24, or even hour 48. Instead, there's a layered, overlapping sequence. Hormonal shifts here, cellular signaling changes there, a fuel source transition happening gradually rather than instantly. Each piece calibrated by decades of research from Cahill's foundational starvation studies in the 1960s, through to controlled trials and mouse studies published within just the past couple of years. Perhaps the most grounding takeaway from all of this is that the body isn't struggling or improvising during these 48 hours. It's executing a plan that's been tested, refined, and carried forward across an almost unimaginable stretch of human history. The specific numbers, hour 4, hour 12, hour 24, hour 36, hour 48, are really just checkpoints along a single, continuous, remarkably well-organized biological story. And that story, as this video has hopefully made clear, is a great deal more intricate, more surprising, and more scientifically fascinating than the phrase "not eating for 2 days" ever quite captures. If this changed the way you think about what's actually happening inside your body during those first 2 days without food, that's exactly why videos like this one get made. To take real peer-reviewed research and turn it into something you can actually follow and remember. If you found value in this, hit like. It genuinely helps this kind of science-based content reach more people who are curious about the same questions you had coming into this video. Subscribe if you want to keep following this series as it continues exploring different organs, different time frames, and different corners of the human body's response to fasting. And if you know someone who'd find this as fascinating as you just did, share this video with them. It's the simplest way to help this channel keep making this kind of deep, accurate, research-backed content. Thanks for watching all the way through, and I'll see you in the next one.